bring in pathogens to bring in white cells to control pathogens. So there are a number of these toll-like receptors. Some are specialized for recognizing the nucleic acid of viruses. Many are on the cell surface are specialized for recognizing bacterial and fungal derived products. But once they set off the alarm with these inflammatory cytokines, they bring the white blood cells that are normally just circulating around in the bloodstream and bring them across into the tissue to form collections of white blood cells that you may have experienced if you've had an infection as pus, where you have this intense collection of white blood cells. These white blood cells, called neutrophils, are particularly professional killers for eliminating bacteria and fungi. Compared to just a generic phagocyte, they are really about 100 to 1,000-fold more effective at killing. Now, there's another system of the innate immunity that's very old and important, and just to show you the diversity of mechanisms. So this one involves tagging the pathogen for destruction by the phagocyte. So the way this works is that there are proteins in your blood called complement proteins. So this is, we all have substantial levels of these proteins all of the time, and they're constantly, slowly being cleaved into smaller proteins that tend to stick to the surfaces of cells. It can either be the pathogen if it happens to be around, or your own cells. If it happens to be on a pathogen, there will be this amplification reaction to make more and more of these cleaved products, and this also helps attract phagocytes to the site of where this is being generated. And at the same time, you also get the ability of the phagocyte to take up the pathogen much more effectively. So this is called opsonization. And the way I like to remember it is this is quoting directly from George Bernard Shaw, The Doctor's Dilemma. Opsonin is what you butter the diseased germs with to make your white blood corpuscles eat them. And that, in fact, is what complement does. This complement protein, when it's tagged on the pathogen, there are receptors for it that make it much more efficiently taken up by this phagocyte, leading to the end of the pathogen. And again, another piece of the complement proteins that are cleaved help attract more of these professional killers to the site of the infection. So you get both of these effects working together to help control infection. There's also a nifty thing that the complement system can do. It can actually make holes in bacteria that where if the reaction is allowed to go to completion, and these holes will actually cause the bacteria to swell up and burst. Now, why doesn't this happen to your own cells? The reason is that all of your cells, they do get tagged by the same complement proteins, but you have multiple proteins on the cell surface that actually are there for one purpose and one purpose only, which is to inactivate the complement system. Most pathogens haven't figured out a way of doing that. So this is one of the most primitive ways of distinguishing between yourself and something that's not yourself that might be a pathogen. There's also recently discovered something called an inflammasome. So there are certain pathogen-derived products that may get inside cells, and this is another alarm system similar to the toll-like receptors that will produce inflammatory cytokines that will help bring in more white blood cells. In this case, these also are very potent at inducing fever, which is characteristic, as you know, of infection. Okay, so that's all I'm going to say today about innate immunity, though I think when you really look at clinical medicine, probably more patients suffer infectious consequences from problems with innate immunity that we unfortunately in this country have the luxury of inducing with things like chemotherapy. But adaptive immunity, of course, has really captured immunologists' attention for a long time. We've known since 430 BC that there was a funny phenomenon that sometimes occurred with very serious infections. So in the plague of Athens, people were dying right and left, but it was noticed at that time that if someone was lucky enough to recover from the plague, they could then go into the population of people who were already, were afflicted and not worry about getting the infection again. So there was something about, there was some process that was allowing there to be long-term protection if you survived the initial infection. And then, of course, more recently, it was discovered that you could use vaccination where you vaccinated with cowpox, a relatively mild disease found on cow udders, and you could then protect people from smallpox. And again, the protection seemed to be, for at best one could tell, close to lifelong. So one of the key things about adaptive immunity is that there's a learning process, but once the lesson is learned, there's durability of the immunity and durability of the protection. And we now know that there really are striking differences between adaptive immunity and innate immunity. So first of all, innate immunity, as I've told you, is ready to go right away. It's on all the time. Unfortunately, for adaptive immunity, it takes probably about 5 days for you to develop the kinds of responses that are characteristic of adaptive immunity. And unfortunately, certain infections may win the upper hand. You may die of Ebola virus infection or of meningococcemia before your adaptive immune system gets a chance to protect you. The adaptive immune system, unlike the innate immune system where I told you that it's going to be the same the first, second, third, nth time, it actually changes with repeated exposure. It gets better and better if you get repeated exposure. And the innate immune response is pretty much limited to the number of receptors you have of these total number. Toll-like receptors, inflammasome receptors, and it's still a finite number. It might be 100 different tricks total in the body in terms of dealing with infection. Yeah?
Just a question on innate immunity. Is it constant throughout your life, or is it not as good when you're born, kind of peaks and then drops off?
So the question is innate immunity constant throughout your life, or are there differences with age? There are some subtle differences with age, but they're important ones. So in newborns, it seems to be somewhat slow to get started, and then you kind of go through this honeymoon period where probably from childhood well into being elderly, it's pretty constant. At the extremes of age, you may actually see again some decline in innate immune function. But again, it's relatively subtle. Yeah.
The people who survived during the plague that was described initially 400, what was the feature that allowed them to survive when so many were dying off?
Well, it would be great to know, but I think part of the problem with the plague of Athens is we don't even know what the actual microbe was that caused it. So we really would only be speculating. So that the question was what allowed some people during the plague of Athens to survive and others not. But even in current, if you will, modern plagues like Ebola virus outbreaks in Africa, we often don't understand why there's such variable courses. There are some immunologists that like to believe everything can be explained by genetics, perhaps, but I think we still have to, prove that. I think the thing that's amazing, yeah, go ahead.
For innate immunity, what about these so-called immune enhancing things like vitamin C and echinacea? First of all, do they work? And secondly, what do they do?
Okay, so the question is whether there are, do these so-called immune enhancing agents like vitamin C, echinacea work? Well, all I can tell you is that most of them have very subtle effects if you look at the evidence kind of based medicine and do immunology, I think they have a fairly modest impact on innate immunity. They may have some moderate benefit, but probably not in the bigger scheme of things anything like, for example, the impact, for example, of whether you're on an immunosuppressive drug or not. It's nothing like that level of impact.
And isn't it true, David, that controlled trials of vitamin C and echinacea have shown no benefit?
Yeah, controlled trials, so Dr. Pisa was asking what's seen in controlled trials. I have not seen any, evidence that there's a significant effect statistically. So the important thing, though, about adaptive immunity is it's highly tailored to the specific pathogen. So if you if you're one of those unlucky people, you travel to Africa, you get Ebola virus, it will come up with an Ebola virus specific immune response. If you never encounter Ebola virus, it will come up with something else depending on what you encounter. And one of the great mysteries for a long time was, how is this specificity achieved? So I'm going to try and get to that. And I think, again, one of the reasons that you really needed the development of adaptive immunity, as multicellular organisms live longer and longer, there was a greater chance that you were going to have repeated infectious threats, that it would be an advantage to respond to, and then in the event that you got a second bout of this infection, you could deal with it much more effectively. So particularly with vertebrates, where now lifespans were now approaching years to decades, you could see that there would be a clear advantage. So the adaptive immune system involves the generation of many more receptors that are specific for what we call antigen. That is, these are things that turn on the immune system that are derived from the pathogens. Literally millions of receptors to deal with any possibility of any kind of infection that you're going to encounter. The adaptive immune system doesn't know tomorrow what that infection is going to be. So its approach has been generate receptors for everything.
David, could you, not everybody will know what a receptor is.
So a receptor is something that can bind on the surface of a adaptive immune cell. There's two kinds called T cells and B cells, and I'm going to talk about both of them. This can actually bind the antigen, which is, again, some kind of product from the pathogen, and turn that cell on. It delivers a signal to the inside of the cell telling it it's time to get activated, it's time to divide, and it's time to carry out an immune response. So to generate these receptors, there's a unique pathway that's called VDJ recombination. The name's kind of a lousy name, but we're stuck with it. It stands for variable diversity and joining, but those are just the name of the segments that are used to make up these receptors. And this is unique. This system only works in two kinds of cells, T cells and B cells. It has no other role in the body other than to generate the adaptive immune system. And what happens, and you're going to learn more about stem cells, I guess, at the end of this quarter, they can give rise to these early T cells or these early B cells. The T cells are called T cells because they develop in an organ called the thymus, which is in the neck. And they undergo this process of VDJ recombination to generate literally millions of different receptors. Each cell has a different receptor. The B cells do the same thing, but they're called B cells because they differentiate into the mature B cells in the bone marrow, so B for bone marrow. But the same process is used to create literally millions of different receptors that can see anything that the immune system is going to be thrown at it. How does this happen? So what these receptors do is you can think of them as sort of a lock and key. This is getting at Phil's question, in that a receptor will happen to have the right shape, if you will, to bind something from a pathogen, to trigger that cell to divide and to become